I want to introduce Pranav Rai, who is part of the IBL team. We work closely with the IBL team. They're the producers of some of the most valuable neurophysiology data I think ever collected, an enormous effort, and a huge asset to open science. So I'm really excited to have Pranav here, who can teach us about this data set and help anyone here who wants to work with it. Thanks, Pranav.
Thanks, Ben. So hi, everyone. I'm Pranav Rai. I'm a research software engineer at IBL, and I joined IBL last year, actually. And the person who hired me actually is also sitting here, Hannah, who was our executive director when we were collecting all of this data and when we published all of this data. And there are, I guess, more people in audience who have used this data set more than me. Yeah, let's go ahead. So I'll go a little bit into the history of IBL. It started as a collaboration between different labs in 2017. And the idea was that the genesis of the project was we have different labs who are collecting different data sets in different brain regions with different tasks. So just to standardize all of these things in a single task and be able to collect data from all over the brain to have a single data set that we can use to analyze and make more reproducible scientific discoveries, basically. So there were 10 theory and 12 experimental labs, which were distributed across 6 countries. Around 70 researchers and 10 permanent staff members. This number kept on going from up and down as people came in. Mostly the staff was mostly research software engineers and some admin staff. And yeah, as I said, the aim was to do scientific research which a small single lab cannot handle and which requires a big scale. And to do so in a more distributed and collaborative way to speed up the process as well. So this was both a scientific project as well as a sociological experiment to see how different labs, which are in different time zones, how they can come together and work on a singular goal. So here I'm just showing where the geographical locations of different labs that were involved. So all of these were part of North America and Europe across different time zones, as you can see. So I'll talk about our data set related to the Brain Wide Map project and introduce the project a little bit here. So this was the main project for IBL with the aim to understand decision-making in the mouse brain. So the idea was to systematically record throughout the mouse brain during a single visual discrimination task. And these experiments were performed across 12 different experimental labs, which had a very standardized experimental setup and procedures with well-documented resources that even if a new lab wants to do the experiment and record a different region, they can do so on their own. And yeah, this was the time when neuropixel probes also were coming in the picture. So we used neuropixel probes to record activity at a single cellular resolution. So here I'm showing the recording plan of different probes that we recorded in the brain. So these are all coronal slices from front to back of the mouse at 5 mm distance from the front to the back. So you can see that some of the regions were recorded on the left hemisphere, which is mostly this last 3 rows, and mostly cerebellum and medulla was recorded from the right hemisphere of the brain. The idea was to maximize recording from areas that share some kind of anatomical connections, and that's how we came up with this grid. And we replicated each recording site in at least 2 labs to maintain reproducibility. And in addition to this, on the bottom, I show another coronal slice. So where we did repeated sites recording. So we recorded from the same region in different labs and try to compare the results from them to see if we are at least getting the reproducible recordings from all these different geographical labs. And in addition to this, there were some recordings that were done in both hemispheres to basically study the interhemispheric interactions. So I'll describe the task now. So on the left, we have our experimental rig setup where we have a head-fixed mice and there's a screen in front of it. And on the right side, I'll show how the task goes. So there's a mouse, it has a wheel in front of it and a screen in front of it. And a stimulus, which is this Gabor patch, it's like a visual grating, which appears either on the left side of the screen or the right side of the screen, along with the GoQ tone. And the aim of the mouse is to move it to the center of the screen using the wheel. So if it moves it correctly to the center of the screen, then it receives water as the reward. Otherwise, there's a white noise that comes up if it performs the task wrongly. And below I show the timeline of a single trial. So initially there's a quiescence period of which can be in between 400 milliseconds to 700 milliseconds, in which the mouse has to hold the wheel still. And then the stimulus will appear either on the left or the right of the screen along with the GoQ tone. And then the mouse has 60 seconds to basically move the wheel to the center of the screen. And if it performs the task correctly, it will get the reward as water. And if it doesn't perform it correctly, then there's an error timeout and a white noise. And this will go on for 2 seconds. And then another trial will start. And this will sort of keep on happening. So there are different sort of contrasts of the stimulus that are shown to the mouse. So the 100% contrast, there are 100% contrast stimulus, and then there are some with 25%, 12%, and 6.25%. So these vary between trial to trial. And then there's a 0% contrast trial where basically nothing is shown on the screen. And the idea for this 0% contrast trial is to basically study the prior expectation the mouse has when it's doing the task. And I'll explain a bit more on this in the next slide. So below I'm showing like one session of the recording. So initially there is around 90 trials where the probability of the stimulus coming from left or right is the same, it's 0.5. But after that, we change this probability to the stimulus appearing on the left side can be 80%, or in some block of trials, it can be 20%. So block means basically a collection of trials where you have different probabilities of the stimulus appearing on either side of the screen. And so you can see that if a mouse is in a particular block, the mouse doesn't know exactly, like there's no indication given to the mouse about that block has changed. So it is just performing trial by trial all these tasks. But we can study for the 0% contrast trials whatever the history that mouse remembers about that particular block, and maybe takes decision based on that or not. So this, what I described before, was active part of the trial of the task. And after we are done with this active part of the task, we have another three more protocols. So the first one is the spontaneous activity. In this, we don't provide any reward, and we keep the mouse wheel as fixed as well. And there's blank screen, and we just record from the mouse brain to just study the spontaneous activity that is happening in its brain. I think this happens for 15 minutes, for around 15 minutes after the main task is finished. And then we also do a protocol for receptive field mapping, where we are trying to map visual cortex by showing different patches of this stimulus to the mouse. And also, after that, we do a task replay where we are showing the same task that the mouse just did, without providing it with the reward or it having to move the wheels. So these are just helpful in getting useful controls when we are doing the analysis, which I'll come to in the next slides. Just to explain a bit more on the experimental setup. So, there's a mouse. We have 2 neuropixel probes that we record from, and there are 3 cameras that are set up. So there's a wheel in front of the mouse that it can move. And there's a left camera to profile the left side of the mouse. Basically, it's to see its left whisker movements in the left side of the face or the pupil diameter. And then there's a body camera which is on the top of the rig, where you can see the movement of the torso or the tail. Here I'm showing 3 trials. So this is like trial number 242, 243, and 244. So on the top, it mentions where the stimulus appears on the screen. So either on the right, on the left, and the choice that mouse made. So in all these 3 trials, it performed correctly. And you can see, so this dashed line is when the stimulus appeared, and this is when the mouse decided to make the choice. So before this stimulus appeared, it had to keep the wheel still, and that's why the wheel velocity is stable. And after that, when it's making the choice, you can probably, I don't know if you can see it clearly, there's a sharp movement in the wheel velocity. So we have all this data set that we can use to make these plots to get the whisker behavior and the licking behavior. And on the bottom, I'm showing the spike sorted output of this data across different brain regions. I'll talk a little bit the hardware that we have used. So we used a BPOD state machine to sync our data and to record our data, and the Bonsai setup. So this is basically listing what kind of information that we record during a trial. So we record like trial start time, when the stimulus appeared, when the wheel movement occurred first, when was the reward given to the mouse, what was the feedback type, etc. And all of this information is present in a trial table that I will come to in the next slides. And for the video data, so these are the 3 videos that we capture on left, right, and the body image. So we track 4 points in the eye of the mouse and get the pupil diameter out of it. We capture the tongue and the paw movements as well. And this is where we are capturing the motion energy around the whiskers of the mouse, which is what you saw in the previous figure as the whisking. And the body camera is used to basically see the tail movement or the torso movement in terms of motion energy. For the EFIS data, we spike sorted the data using PyKilosort 2.5, and we also provide some kind of features out of this EFIS data as a part of our output data sets. These features are related to the root mean square of the AP band and the LF band, as well as some power spectrum features in specific different bands of the like alpha band or beta band of our recording. So because all this data was being collected in different labs, we had to maintain a central server where we aggregated all the metadata for our experiments. So this we called Alex. This is also like an open tool which any lab can implement if they want to if they are collecting data and to maintain the metadata related to their experiments. So it captures your subject, start time, end time, all different metadata related to the experiment that you have done. So here are some numbers of the final data set. So we recorded from 12 labs. There were 139 mice, 459 sessions, and we recorded from 699 probe insertions in around 281 brain regions. The total number of neurons that were output from spike sorting was 620K. But after doing all the quality control, the final number of neurons that we got was around 75,000 good neurons. Yeah, here I'm showing the neuronal yield per brain region. So on x-axis you have different brain regions, and the bar shows the number of neurons that were recorded in that brain region. And the filled part shows the number of good neurons. So this is around 10% because the y-axis is like logarithmic scale. So it's basically showing that different regions had different neuronal yields. This is because we did equal spaced grid when we are doing the recordings, and some regions are bigger, so more number of neurons were recorded from that. And we try to make plots like this. I'm showing on the left bottom corner of the screen. This is called a Swanson flat map, where we try to basically, for any variable that you want to show for a particular brain region, you can plot it in this kind of map to see everything in a 2D image for what's happening in different brain areas. And we chose a sort of brain hierarchy where we are selecting for 10 regions. These are isocortex, thalamus, hypothalamus, midbrain, and other main brain areas. So it's not as granular as LN-CCF brain hierarchy, but it was relevant for the analysis that we were doing, basically. So the main, I'll describe briefly the main result of the Brain Wide Map paper, where we basically try to decode these different task variables, like the stimulus, where it appears on the screen, and the choice, whether the mouse decides to do a left turn or a right turn, or if it was rewarded or not, and the feedback. And we try to see how much information is contained in the brain regions, in different brain regions for these task variables. And we do this kind of decoding analysis, and what we see is, like, for certain task variables like stimulus, the brain regions that are encoding it are very few, like it's only 40 out of 200 brain regions are coming out of significant, while other task variables are encoded throughout the brain. And we do, instead of doing just a single decoding kind of analysis, we do multiple analysis on this data using single cell analysis, as well as population trajectories and different encoding models. And we it sort of gives different ways to look at the same data. And yeah, I'll briefly go over the analysis that we did in a more simpler version in the next few slides. Yeah, so regarding data access, internally we use another API which is called ONE API. So this was before when we started, there was not a big NWB ecosystem, let's say. And we have been using it for all our publications. But this year we've also released all our data set in NWB format, and people have already talked about this. And this is, I think, is a great resource for other people to directly interface with the data. Yeah, and for this NWB data set, it's like around 50 terabytes of data. there's a lot of files that you can see. It contains the behavior data, video data, and the Neuropixels recording data. Ben's team has immensely contributed in trying to convert all of this data into NWB format. And on the right, I'm showing basically how this data is organized for one session for a processed NWB file. I think there are better ways we have seen today. I just put something before. So we have subject information, devices information under the general field. And then you have different epochs, which tells you whether it was a passive time block or active time block. And then different variables related to trials, like when the stimulus appeared on the screen, what choice did the mouse make, etc. And then we have unit level information, which is basically what were the spike times, what were the IBL quality control score, what was the firing rate, mean waveform. So there are some notebooks which are part of this DANDI notebooks archive that Ben had mentioned in his talk. So there's this one, which is the BWM usage notebook. I'll just glance through it. So this was made by Ben's team, I think Heberto. I think it's a great way to just explore the data and see different fields that exist in the data set. So yeah, this is information related to electrodes and which areas they lie in. One thing that I really liked is they had a cell related to directly streaming the video of the mouse in this notebook. So yeah, you can basically, I'll go through this notebook to explore the data set more. Another notebook is, it sort of does, I think it's here directly. It does a lightweight version of what information that you will see in the Brain Wide Map paper. So here, basically, we are instead of aggregating the data over all the sessions, I've just chosen one session, and we are trying to replicate some of the analysis that was done in the paper. So I'll briefly go over this. So yeah, this is the epoch table that I showed in the organization. So you can see this active part of the trial and what is the start and stop time of it, and then the passive protocol and the start and the stop of start and stop timing for that. There's trial table which lists all this information about when does the stimulus appear on the screen, what choices did mouse make, whether it was rewarded or not. Yeah. And then there's unit level information, basically the IBL quality control score, whether it was a good unit or not, and the firing rates of this particular neuron. So for one of the analysis, which is basically trying to do a single cell, whether to assess whether a single cell preferentially fires more or less when mouse moves the wheel to left or right. So this is one of the analysis that is present in this notebook, where we first try to get similar number of trials for both when a mouse performed a counterclockwise movement versus a clockwise movement. So in this case, it's around 300 trials that we select. And then for each neuron, we try to get this kind of modulation index, which is the firing rate when the mouse did a counterclockwise movement minus firing rate when it did a clockwise movement, divided by the sum of these two. So if this value is a large value, then it means the firing rate is very high when the mouse is making a counterclockwise movement versus and vice versa. And so we can basically wrangle the data to get to a data frame, which I'm showing here, where you have different neurons as columns, and on the different rows are different trials, and we are storing the spike counts for each of the neurons for this trial during the period before the mouse decides to do a counterclockwise or clockwise movement with the wheel. And this is the choice label, which tells us what choice the mouse actually made. Yeah, and based on this, we can group our data to get a left side firing rate and right choice firing rate, and calculate the modulation index based on it. But we need also a control to see how, if it's just by chance that we are getting this number. So what we do is we shuffle this choice label I'm showing one example shuffle where we are basically randomly assigning counterclockwise or clockwise in these different rows. And then we again calculate our modulation index based on that. And we do this kind of shuffling process around 1,000 times, and we'll get a p-value based on this distribution, and we can see where our modulation index for the actual data lies there. So you get something like this, where the orange means the results were significant based on the p-value versus not. So you can see that there are some, so on the x-axis you have different neurons, and on the y-axis you have the modulation index. So you can see there are some neurons which are preferentially firing for the left side movement versus the right side movement. So there's some information which is present in the single cells. So this is a very lightweight version of the analysis. In the paper, we put a lot of controls to do this analysis and more sophisticated statistics to try to make a stronger claim regarding it. But this notebook basically, does similar things in a more simpler way. If you want to just get started with using this data set. Yeah, so these notebooks are present as a Colab notebook, and I can share with the people here. And if you, and these are the two main publications that came out of our work. So the first one is just described into more detail the kind of analysis that I was showing, and the second paper is regarding the prior information that mouse uses. And the brain-wide representations of that prior information. We also have a visualization. So I'll describe briefly different tools that we have also developed along the way. So there's a visualization website that we have developed. I'll show a brief demo of it. So I think there are similar things that exist in DANDI as well. So here we have a search bar at the top where you can either specify a session ID, or you can specify a location where the recording was done, or even a region, a brain region, for example, here in this case CA1, and then you can select a probe and it will update the right side metadata for that session. And initially we provide some kind of session overview where we have some spike rasters and different sort of recording snippets from the session. And then there is some behavioral level description, different psychometric curves, and how the paw movements were there, for example. And then we have trial level information where you can go through different trials and see how the mouse was performing during that trial. And there's also a video that you can see as the mouse is doing the task. And the panel here will update accordingly with the time as the mouse is doing the task. Yeah. Yeah, there's some more information related to the neuronal level. So there are different clusters, and you can see different firing rates, different waveforms that are recorded on the electrodes on the right here. So yeah, and you can see the relationship between this and the stimulus on time and first movement, for example. So it's similar to NeuroSift, I think that Ben showed, but this is more like curated version for IBL dataset, because NeuroSift is a general tool that works with everything. So this is also a great way to explore the IBL data. So I'll briefly mention other projects that are going on in IBL. So one is the EFIS Atlas project, where we are trying to see how the neuronal activity can be used to infer the brain region from which it was recorded. There are other modalities that we are working in, for example, mesoscope, wide field, and fiber photometry, which are basically doing the same version of the task, but in mesoscope we are using imaging to do the analysis. So some of these are still going on, but the hope is to release the data for this also in DANDI by next year. Yeah, I'll describe, so how much time? Maybe 5 minutes. I'll describe briefly the EFIS Atlas project. So in the Brain Wide Map paper, we had around 700 insertions, but we had to remove some of the insertions because the behavior of the mouse was not up to the mark, did not perform all the trials that we needed it to perform. But still, the information, even if the behavior was not correct, we can still use the EFIS data from those recordings and try to, if our aim is to just relate the brain region to the EFIS recordings, we can still use that. And so by this, we get 400 or so more recordings that we can use in this project. And the aim here is to extract some features out of the raw data, like this RMS AP, the power spectral density in different bands, and some spike detection features. And for example, here I'm showing the brain region according to the Allen hierarchy. So you can see that there is high power, for example, when the region is transitioning from hippocampus. This green one is hippocampus and this pink one is thalamus. So we can use this kind of information to sort of judge different brain regions just from the EFIS data. So we have built some tooling around it, the way to calculate these features and to do it at scale, for example, in a supercomputing cluster. Some kind of the visualization that you see, we have a visualization website for like this for different sessions. And one thing we can do is we can build some kind of a reference atlas where we can show for each of these regions what is the average or mean of that feature and how it varies across different regions. So there's a visualization website for this as well. You can see, for example, this. You can scroll through different features and see how they differ in different brain areas as you go through front to the back of these coronal slices. Another thing that you can do using this kind of data set is try to decode the brain region. So here we can use this and fit some machine learning model out of it. Here I think we are using XGBoost and make some kind of prediction out of the brain region. And we see that it's very close to the actual brain coordinates of these channels. So I think we reach something like 70% accuracy for these kind of simpler models. Not actually, I think it's more like 62%. With more complicated model like transformers, we reached around 70% accuracy. But one thing I was thinking of trying here was trying to gather more data set from DANDI and try to get a bigger data set and see if it improves our model capabilities. And as a last item, I'll also mention about, I think many of the labs are trying to build some kind of scientist in the loop analysis for themselves. So we have given a shot at it as well, something called IBL AI agent. So the idea here is to use coding agents like Codex and Cloud to analyze IBL data. So what we do is there's a repo for it, and you can clone it and then use it along your favorite LLM agent. And then you can chat with it to come up with analysis and hypothesis. So we have different skill files that we have made for this IBL AI agent, along with some instructions specific to IBL data. So if you ask a question, it will try to convert it to a very focused question, trying to pin down the analysis that it will perform in a very interactive way. So for example, the analysis that I was showing you, it will try to ask what is the bin size that you will use, what is the time window that you will use, instead of trying to do everything on its own. And instead of, because it takes a lot of time to individually download each session, we have built like a compressed data, which has the spike information and behavior information and other key points information. That is, I think, in a compressed form, it comes out to be 10 gigabytes. And that is downloaded along with this agent. And we can use this to do our analysis more quickly. So yeah, it sort of, the agent is designed to make some kind of rigorous workflows, and it takes out some data as a held out data set. It performs all its analysis on the rest of the data, and then finally uses the held out data set to test whether what it's testing, whether what we are analyzing is significant or not, for example. Yeah, so this is still a work in progress, but I think it's also a good way to inspect our data. So currently it doesn't use the NWB ecosystem, but yeah, if someone wants to try it, it will be useful as well. So this is the repo link. It's IBL-AI agent. You can give it a try. And the main contributors to this are Cyril, from people from our staff team, Cyril Rosant, Olivier Winter, and one of the PIs, Kenneth Harris. So as overview, yeah, we have our brain wide map data with 609 neuropixel recordings. This is the DANDI ID for it. We have both processed data as well as the raw data for it in the DANDI dataset. And in future, we'll include the data for EFIS Atlas and other modalities as well. And with that, I will want to acknowledge a lot of people who are involved in this project and our funders, Simons Foundation, Wellcome Trust, and NIH. And briefly, I want to mention, because this phase of IBL 1.0, it sort of is finishing, or it finished with the release of our two main papers. And in the new phase, we are a group of research software engineers, and we collaborate with different labs to make the to collaborate with them on their scientific research. So the idea is that the different labs will collect experimental data, and we provide support in terms of all the capabilities that we have built related to processing and data analysis and visualization tools. So yeah, we have worked through a lot during the IBL 1.0 phase to work on our data access protocols and data processing pipelines. And we also do a lot of community engagement and outreach in terms of workshops like this, so which can help different labs publicize their data set as well. And we build some kind of web-based tools like the visualization tools that I was showing before. And of course, we partner, we and our partners are committed to open science and releasing all our data and tools. So we request proposals from different labs each year. We have already started working with some projects, some labs this year, and we'll call for a next request of proposals next year. So please follow for our social media links on X and LinkedIn for these requests, and if it's for interest for you. Yeah. And with that, I'll take some questions if you have.
Just curiosity how it's written there, but then so that you're using just for the issue.
Yes, we use NeuroPixel 1.0 for these recordings, which is the longer one with 384 channels.
Okay, so it's not mixed with the new technology of NeuroPixel 2.
No.
It's part of maybe the new phase, like. Or?
So there are some projects, which is a project called Multi-Area Project, which uses NeuroPixel 2.0, which is this 4-shank probe. But this project, I think, is still going on, and probably we'll release data for it also by next year. But I think it's also publication ready in terms of it.
And just another curiosity about the simultaneously recorded different area per each session. In the sense that across all the recordings, you have a huge amount of data. But like, the average amount of like triplet of area or quadruplet or?
Average amount of triplets, like in terms of brain regions that we have recorded from. I don't know by heart this number, yeah, but we should be able to get from the DANDI dataset. Yeah.
Thanks.
Cool. On time. Yes.